All right, good morning, everyone. I'm Jacob. I'm going to talk about how to use data pipelines in machine learning for material science. I'm a PhD at UCL, University College in London. I've also applied my research at Spotify, so your music sounds better. And since 2018, I've been working also in material science, working at the Army Research Laboratory on epoxy glass composites. Nowadays, I spend my time on something called Matterhorn Studio, which enables instant access to machine learning pipelines that are cutting edge and they're really great for lab automation. And so today, I'm going to talk to you about lab automation, why we do that, then why we need machine learning for experiments, and then ultimately, why we need pipelines for that machine learning. All right, so materials do rule the world. You're sitting on chairs that are made of materials, the computers made of semiconductors, and right now we need to kind of figure out how to make these materials in a more sustainable way that can be recycled. And biosynthetic materials are quite attractive for that. So for example, with yeast cells, we can actually produce fuel. We can also produce beer, which you might know. And there's also biosynthetic materials that can help with drug and pharma production lines. So, of course, we want to maximize the output from these yeast cells. So what kind of processes can we go with that? And so, for example, here's a process at the Technical University of Munich. And they're working on these yeast cells. They have a whole bunch of complicated steps from discovery to scale up. But today, we're just going to talk about that middle part here, which is a loop where we have some yeast cells and we're going to feed them vitamins and trace minerals, the same way you kind of feed things to humans to make them strong and produce better science or more fat or something. So why do we do lab automation and research, for example, for yeast cells? You see here some machines. There's an OpenTron, costs about $10,000, up to maybe a Beckman for $500,000, and then custom designs for multiple $1 million. And the great thing with these lab automation systems is they give you high throughput, so they just produce a whole bunch of data. More importantly, they're also reproducible. So instead of having 3 scientists do the same experiments and getting 5 different results, you have basically a robot that will do it almost the same way every single time. And so that leads to the scientists being freed to just do the science, which is what they sign up for in the first place. Now, specifically, what you want to do in these kind of setups is that you have a closed-loop experimentation system. So for our yeast cells, for example, we're going to have a first step where we're just going to grow them and feed them stuff. That takes maybe 18 hours, and then you're going to move them into a measurement stage, so step 2 here. And then finally, when you have the data, you have to kind of make the decision of what do I do next? What do I tell the robot to run next on the experimentation machine? And so this automation is actually happening across all kinds of disciplines right now, physics, chemistry, biology, who are trying to figure out better alloys for faster aircrafts, chemicals, paints of combinations, plastics that are more stable but more sustainable, and concrete that doesn't require, a kiln firing, which is really, really energy intensive. So here you see different robot systems that have been put together over the last 5 years. Now, why do we need machine learning to run these experiments or to plan specifically these experiments? And the central question there you want to ask is, what experiments should we run next? And so in this example here, again, we have our yeast cells. And so we're going to look at just temperature, just one parameter. And so we maybe look at anywhere between 10 and 30 degrees. And so right now we have some historical data points here. So these black axes are previous experiments we ran in the lab. And right now, 25 degrees is the best top runner. It produces most of the lipid yield, so the cell performs best at 25 degrees. And in the middle, we're missing data, right? So what should we do next? How should we just explore this kind of parameter space? The first one kind of strategy is random. So you just throw some dice, or you throw some, if you're bad at darts, I guess you throw some darts at the dartboard, and you just hit some different temperatures, you put them through your lab automation setup, or you just do it manually in the lab. And it's actually a statistically valid method. It's just not particularly efficient. And so maybe you want some more structure. So with a grid approach, you just kind of chunk up the parameter space in even spaces, and you just explore it that way. So that way we will get a better structured idea of how these yeast cells behave. And then finally, we have design of experiments. There's a show of hands who knows design of experiments. Wow, okay. Different crowd. Right, so design of experiments is the established method to run experiments, for any kind of experiments, really. So specifically, of course, we want to characterize the behavior of any kind of natural process. But for example, with these cells, we might think that there's some kind of red line that characterizes that behavior. And design of experiments helps you to pre-calculate which experiments you need to run and to get the best kind of line of fit. Now, we're moving into the stage of machine learning nowadays, and so there's actually a fourth method now called Bayesian optimization. Let's try this one. Bayesian optimization, show of hands. All right, that's a bit more. Good, I'm happy. So I'm going to explain to you Bayesian optimization now in 3 minutes. And basically, here's a red line, which is kind of nature. So this is the true yeast cell behavior. Of course, nature never tells us, and we kind of have to play with nature and run those experiments to get some data on the yeast cell behavior. So obviously, I just created a red line. I know the truth now, so I can show it to you in this example. So here we have some historical data again, and we can fit a machine learning model here with the blue line. So the blue line is guessing right now quite well what the data is, but then in the middle part we don't have data, and so the guess is kind of regressing just to the mean of what it sees in the other data. And on top of this specific machine learning model here called a Gaussian process, we also have an uncertainty measure. So you see that confidence band is quite tight around the places where we have data points. And then where we don't have data, so in the middle part, it's quite wide. So that means machine learning model says, I don't really know what's going on, don't trust me too much on my predictions. And so with the predictions and the uncertainty measure, we can now apply kind of acquisition functions or expectation gain functions that will help us decide what experiment to run next. So that looks like this here. So we have this orange line now. That's the mathematical calculation of the maximum statistical information gain for this specific point. And so right now the machine learning model thinks that running actually around 25 is a good idea. And that kind of makes sense because 25 is the highest performing one. So let's explore a bit more around this space. So we go in the lab, we run the experiment, we put in the data, and so this is our new kind of understanding of the world of yeast cell behavior. And so, again, the expectation measure here in orange now suggests for us to move closer to maybe 22. So you go in the lab, you run the experiment, you put in the data point, and now we see we kind of split up the space already in a high uncertainty region on the middle left, and then another one on the middle right. And so a machine learning model is getting a bit funky here with the blue line, and it's suggesting that we actually go back to the 25 degrees. So we go back into the lab, and we explore that. And now we put in the data point here, and now the machine learning model, as you can see, is slowly squeezing out the uncertainty in an efficient way. And furthermore, we're actually moving to the optimal point at 18 degrees Celsius. So a few more steps. There's 18, there's 19, there's 16 Celsius, and then finally at 18 degrees Celsius, we have reached the maximum point. So machine learning with optimizing around these uncertainties eventually will converge to the optimal temperature. Now you're going to tell me like, well, Jacob, I can see this obviously, it's right there. It's a temperature and I just explored. But the fact actually is that in high dimensional spaces, Bayesian optimization will just be a better choice to explore these things. And so especially when scientists need to deal with 4, 5, 6, 7, 10 dimensions, it is much better to use these kind of methods that are efficient to search this space. Okay, well, I'm glad we got through this. You can try this again on Matterhorn Studio and play around with the demo I just gave to you and get a bit more experience with that. Now, here's an overview again why Bayesian optimization is a good choice. Specifically, random is simple, but it's not particularly smart. With the grid, we have a bit more structure, but it's just too expensive on high-dimensional spaces. Design of experiments is a great choice to make your interpretation and make a first step. But it's complex and not particularly efficient in a lot of applications. And with Bayesian optimization, we're able to most efficiently navigate the space. But it's also complex and it's data hungry. So data hungry, well, we just kind of try to produce most data, for example, lab automation. And then the complexity part, we're trying to fix that with Matterhorn Studio. So that's the little sheep here. So why do we need data pipelines for machine learning is the final step here today. So there are challenges in Bayesian optimization, of course. There's no free lunch. And so the first one is that we need to find the right algorithm. So different machine learning algorithms that are specific and perform better for certain materials. And then you also want to collaborate with an expert on it. So you don't want to leave statistical efficiencies on the table, but you want to integrate them into your machine learning model. And then finally, of course, it's science, so we want it to be reproducible and transparent and interpretable. So it would be great to have some kind of like structure and standard, right, to standardize this. Well, so we put together something called the Opt Store, which is an optimization store. And it's basically just a place where you have access to all of these algorithms. There's a REST API. It's based on Docker containers. And of course, as you can imagine, it's using Nextflow pipelines to structure all these algorithms and put in one central place. And so right now, you can just click on a button here, click on Run. If you set up your data, it will create a draft. You click on Start here, it will go into running mode, and then it will give you the next experiment to run. Either you pipe that into your lab robot with REST API again, or you just go and do it by hand. Now, what's happening in the background here is actually this is just a Nextflow pipeline. So this is the pipeline. You can look it up right now on GitHub. And this pipeline has a few modules in it. And the one that's kind of like, the moneymaker is a Python file where we have, this is a platform called Blowtorch, another platform, a package called Blowtorch by Facebook, and it's a very popular Bayesian optimization method. I just put that into that Python file, and the pipeline pulls the data from our platform, runs the Bayesian optimization, and pumps the result back onto our platform. And that's all just very easily done with Nextflow, because this is the workflow we're going through here. In the Ops Store, you kind of choose an app, and then the Nextflow Tower will just schedule it on the AWS batch, and then that one will run it on an EC2 container, whatever app you chose, and that will, put the result back into the platform. And to support all of these efforts, we kind of created a forum called OptCore, which you can see obviously is inspired by NFCore. And so that place, we're trying to specifically answer questions to machine learning and Bayesian optimization, and how to make these work with the Nextflow containers. So here, example, there's a category, Blowtorch single task GP. That's a specific machine learning model. People just can click on that and share about how they use it. And so, of course, because these are Nextflow pipelines, we can do all kinds of interesting things. First of all, they are reproducible and transparent because, well, you just go in the Git repository, and if you just download and have the same container, it will always give you the same result. Then with the OptCore forum, you can actually discuss these efficiently. And then finally, in terms of collaboration and making sure that as a community we're all moving forward efficiently, you can just fork the pipeline, make your own changes to it, and discuss those results with the community. And I think that's better than the chaos right now that's happening with hundreds of pipelines, hundreds of Python packages being put out there and not being discussed or collaborated on in an efficient way. So putting it all together, so machine learning helps run the best experiments for material science, but in general anyway. With the Opt Store and OptCore, we're trying to help people find the best algorithm and make that run. And then with Nextflow, we've really found a solution that helps us to collaborate in an open science way to make these pipelines work. All right, thank you so much.
So thanks, Jakob. We have, I think, time for one question. Do you have any plans of trying to put any of these pipelines on NFCore, for example, maybe some material science pipeline that you think could fit?
Whether they would be part of NFCore?
Like, for example, you said you have some pipelines, you create OptCore, right? But have you thought of maybe submitting some of the pipelines to NFCore? Just a question.
Yeah, so I discussed it with Phil. I think it's something that I would love to do. It's about how to integrate these communities. We're trying, so our goal is to support people to really get the Bayesian optimization right in the best way. And so NFCore is obviously predominantly bioinformatics right now, but if we could find a space to put these together, and I think it will happen, not just for material science or bioinformatics, it will come to other areas of science too. I think in a way it's just a matter of time until these things merge.
Totally. Great. Thank you very much.
Thank you.